Hello and welcome to Talking Meds, a podcast from PrescQIPP, engaging conversations about medicines-related dilemmas. I'm your host, Jonathan Underhill, Medicines Clinical Advisor here at PrescQIPP. Hello and welcome to the latest episode of Talking Meds. This is the second episode talking to Vicky Chaplin and Paul Selby about Genomics-informed medicines optimisation. In the first episode, we talked about the general principles of this. We talked about the national work that's going on and some of the regional and local work that's going on, the networks that are available, and some of the education resources where people can get up to speed with this should they want to. In this second episode, we're going to delve into a little bit more detail, really, about some of the specifics around some of the technologies that are being used and some of the sort of the key principles around the use of pharmacogenomics and genomics-informed medicines optimization. So, Vicky, just to start with the 10-year plan, because I think we mentioned last time, it's mentioned, I think I said 117, I think you corrected me and said it's 118 times. It's mentioned, 119 times it's mentioned in the 10-year plan, the word genomics. And many of those references are aligned to when they're used with medicines as well, aren't they? So what does the 10-year plan have in for us in terms of genomics then?
So genomics is kind of one of the big bets for the next 10 years. So I think I mentioned last time as well, there's a quote in there which it says that genomics is expected to inform 50% of healthcare episodes by 2035, which is obviously a huge amount, and we would expect a lot of that to be to do with medicines. And I guess underpinning all of that and throughout and the mentions of genomics is about really integrating the use of genomics into routine clinical care. And if we do that, then it will really inform the use of medicine. So there's an element of expansion, I guess, in a lot of the areas where we're possibly more frequently using genomics to inform medicine. So, Paul touched upon it last time. So we've got an really increasing number, which is great, of kind of targeted therapies where we're using genetic results and we're seeing medicines that are kind of really targeted for causes of disease, both in cancer, where we're seeing, loads of them coming through, but also in kind of other inherited diseases, and starting to utilize genetic information to really target the therapies that we're using and to optimize treatment. And I think, there are elements in the 10-year plan, such as increasing the breadth of our cancer panels. So really offering comprehensive cancer genomic testing for everyone will mean, although you may see it on the face of it as a diagnostic or a prognostic test, actually you've got a whole bank of information in there which may inform future medicines use. Same as looking at making sure that our testing is up to date for rare and inherited diseases. Again, we are working towards having as much information as possible for any medicines that may come in the future. And I think also that looking forwards is really in there as well. So, horizon scanning, working with industry, because we've got the life sciences sector plan as well that came out around the same time, and that talks a lot about genomics. So, this year we've started having clinical trial targets for cancer on our panels. So that means that information is already there for that patient potentially in the future. Perhaps it's not in the standard kind of treatments that are available now, but actually they may be eligible for a clinical trial. And how great you've picked up that information as part of their routine testing when they first came in. And then of course there's pharmacogenomics, and that gets its own little special mention, quite a lot of the way throughout. And I guess in terms of pharmacogenomics, so how we may respond to medicines, it's often, traditionally, they were also seeing it increasingly with new medicines coming through where it comes with information on pharmacogenetics. It's often associated with medicines we might have been, more familiar with for years. And we've got lots and lots and lots of people, using those medicines and, potentially wanting those tests in the future. So at that point, we're getting to population level testing. So that's kind of the big kind of headline in the 10-year plan is about having this genomic population health service. And again, it's not a separate kind of, it's about providing genetic testing as part of routine care. And the crux of it, where we're trying to get to, I guess, over the next 10 years is now obviously our testing is a bit more reactive, point of prescribing, or when somebody comes in with an issue. What we're working towards is having that kind of preemptive testing. So you've already had some form of testing done. That information is there. And then so when you go in for your appointment and you need something, that will inform the treatment that you're given. So that's really what we're working towards. It's just, other information on file about you that will help your healthcare professional find a choice with you that works best in that current situation. So I think it's going to be, there's lots of things that are needed that are highlighted in the 10-year plan, like the informatics infrastructure, workforce training and education, all absolutely essential. Again, I say this a lot, it's not just about providing a test, but really exciting. And obviously it's great that there is so much interest and enthusiasm for it across the system.
Yeah, I mean, I get that impression too. I think there's a danger here as well, though, that people think that this is going to be the answer to everything. And of course, it's not, as we talked about last time. The words here are really important. Genomics-informed medicines optimization, or genomics-informed decision making. It helps to give a bit more information, maybe specific to that individual, that can help them and you make a better decision for them. It's not going to fix everything. And as you say as well, there's a lot of moving parts here, aren't there? There's lots that need to happen for this to be realized within that 10-year time frame that's been put on this. So just in terms of specifics then, so we've talked, haven't we, about the different tests that are available. I don't know whether Paul or Vicky, whether you want to take this, but what are the key differences between point of care tests and lab-based tests then?
Yeah, so I think the first thing to differentiate between what the technology is here and how it's used is really the difference here. So with all of the genetic testing, you're essentially looking to take a sample, extract DNA from it, and then run it through a sequencer. That sequence will then produce what we talk about these things very loosely, so please forgive me, but your GCTAs, all those runs of nucleotide bases which make up your DNA, and therefore give you the result that you can then assess what that means for the gene, what that means for the individual. And if you think about, really, there's two things here. Laboratory testing, where you can use any technology within that setting, whether that be a large multi-million pound machine that splits the DNA and runs it several times, and you get really high quality testing. Or we can bring that down and we can look at specific genes. We could alternatively look at the entire genome, or we could look at specific variants within that gene. So once you start to go down to that level where you're looking at a specific variation in a gene, which is known, which we can then use to predict a function, then that can be used to make the sampling a little more simplistic and therefore applicable to a test that can be done at the patient bedside or near to the patient. So that's when we're talking about point of care tests, we're really thinking about near patient testing. And that's because the technology has been developed and has moved on to allow us to do these smaller specific tests. So I'm just trying to get people to think about that difference between, in a lab, you can do massive sequencing, you can identify variation, which allows you to predict that the patient is going to have a specific rare disease, which you've not seen before, or you've not seen that before. Whereas with pharmacogenomics, we're much more interested in those variants which have an evidence of impact. So we can then take specific variants that we would want to look for, the ones with the strongest evidence, and then a manufacturer can essentially turn that into a near patient or point of care test. And the advantage of those is because they're simpler. You don't need the laboratory trained staff. You might need the necessary reagents, and the systems might be smaller and easier to process and report on. So all of that means you can then have this thing where you get a box in a room on a ward where someone can take a saliva sample, run it through the machine, and you get your result back within an hour. So the key difference is really on the setting you use it and how you use it. As you can imagine, getting that sort of quick turnaround time, getting that accessible to the lab is really beneficial when you're looking to preemptively get that result so you can then make a decision for the patient. I think one of the other things I just want to touch on is there's a limitation from simplifying the testing. And that is if you're only picking variants with the highest evidence base, or you're limiting what you test for to make the sampling more easy and therefore practical, you might be limiting what you can find out about the patient. And if you look at the NICE health tech guidance on clopidogrel, and it assessed 3 different systems of assessing the CYP2C19 genetic variation. 2 of the point of care tests provide testing for 3 or 5 of the variants that we know about. There's 35 variants that are widely reported in the literature. And so you've just got to be really clear on what has been tested and what has not been tested as part of implementing that point of care.
Yeah, so I mean, it sounds to me like it's not dissimilar to other diagnostic tests in some ways, where you have a sensitivity and specificity of the particular tests. And as clinicians, we need to use those to work out what the positive predictive value of the test is going to be. So if I get a test result back that's positive, what does that do to the likelihood of the person having the condition that I'm trying to diagnose here? And it's very similar sort of principles here, isn't it? And you mentioned about that sort of the lab standards that are there that you have where there's a lot more information comes from lab testing. And then I think obviously the convenience of having a point of care test or a test on a lab, but there's always going to be a little bit of a sort of a trade-off there, isn't there? Because that's probably not going to give you as much detail or indeed potentially as much accuracy as it would in a lab-based test that maybe takes a bit longer to do and costs a lot more money. So with all these things, there's always a trade-off. There's a trade-off with point-of-care diagnostic tests. It sounds like there's going to be a trade-off with point-of-care genomics tests as well. So no, thanks for that, Paul. I think that's really helpful. Vicky, did you want to come in there as well?
Yeah, I suppose I was just to come in and I suppose from both sides, although lab-based genetic testing is obviously the main one that we do, it's what we deliver through the NHS Genomic Medicine Service currently. And as Paul said, we can go from looking at really specific targets on specific genes all the way up to whole genome sequencing. And I think that's going to be the balance we're thinking about in the future, because we've got research trials as well coming through that we're working with Genomics England on. Looking at doing more comprehensive sequencing for people to potentially pick up more, and it's that balance of looking more broadly to things like turnaround time, logistics, things like that. And I think ultimately it may end up depending on certain clinical situations, but I think the other aspect for me is also adaptability. And this is such a fast moving area. Even if we know that we're looking for variants in a particular gene, the variants we are looking for may change as we learn more and more and more. So in all fairness, NICE highlighted in the CYP2C19 clopidogrel guidance that lab testing could test for more. It doesn't necessarily, and I think that's important. So we have certain assays that have to be available, and those assays can test for certain variants. If we're doing targeted variant testing, they can't necessarily accurately pick up all of them. So the labs have to work on that with companies, and over time we get more comprehensive assays that we can use. But that does take time, and it's not always so simple. And I think it's just really important, particularly, and again, this is in just to mention the CERSI guidance as well. It talks about, I think a big thing for us is ensuring we cover the whole population. So I think most people are probably familiar with the fact that one of the slight issues we have with genetic data is that it is predominantly based on a European Caucasian population. So although we've had evidence that's been around for years, it doesn't necessarily cover everybody in terms of the most frequent variants we're going to see. So that's a huge aspect of work across the whole of genomics, and increasing our understanding and making sure that we're doing tests that are representative of our population, and that we have the breadth in what we're testing to pick those up. And we can see that, for example, with DPYD, having to add in a fifth variant because we hadn't covered that. The original 4 that were recommended, well, actually, there's another one. The evidence came out, so we need to add that in to best serve our population. But that then involves we have to go to the labs and discuss, can you set this test up? What do you have to do? How do you have to change it? So I think adaptability is really important, and it's something as well, I guess, just to bear in mind. It takes discussions with the labs, but also if you have a point of care device, you need to be able to make sure that you can refresh and update that potentially in the future as you need to.
Yeah, absolutely. I'm actually quite reassured by that. When the science changes, the decisions should change. It's when people don't change their decisions when the chance that's when it gets problematic, isn't it? So on that then, in terms of the evidence base and the big trials that are underway in this area, I've heard of the Progress study. I mean, would one of you like to tell us a bit more about the Progress study, and then perhaps about other studies that are underway as well?
Yeah, I mean, so I can kick us off around the Progress study, and then I don't know, Paul might have some insights as well from locally. But in terms of the Progress study from our national perspective, it's really exciting actually. It started off as a transformation project, and then it was taken through in our pharmacogenomics and medicines optimization network of excellence. And essentially, it's an implementation study because there'd been lots of talk over the years about how panel-based testing was best. Most prescribing is done in primary care, all fairly simple facts that are easy to verify. But then how does that apply to the delivery of pharmacogenomics? If you take all those principles and go, okay, well, that's fine, I'm just going to go give a panel in a GP practice. But actually, is that helpful? How does that work? What do we need? So it's led by the Northwest, but essentially they then rolled it out across the country. We ended up with 21 sites. And they just had a few drug gene pairs on there where we were really kind of confident about the evidence base. The GPs were kind of shown how to use it. And over time, there's things that came out of that which really developed, even during the study in terms of how best to deliver this. So, they went from kind of your standard blood test sample to going, actually, let's use saliva because it's a lot more convenient and the patient can take it home and put it in the post. They had, full consent because it was a trial, and, it's not necessarily consent with pharmacogenomic testing like it is with kind of whole genome sequencing or whatever, but having that discussion with a patient, they went to kind of e-consent because that's more helpful. They had information about what to use, for the clinicians in terms of advice, what the test result meant, so they could discuss it with a patient. And that was developed into kind of a little CDF to provide that information. So almost all the way through, they're adapting and learning and getting feedback from the patients and the clinicians who were doing this in order to kind of say, actually, this is a nice idea on paper, but if you do it in practice, these are the things that you need to do to actually make sure it's a proper patient-centered service and that it actually affects medicine use. So they kind of, that trial's like closing down now, and we're hoping that they're in the process of kind of looking at all the results. So that should be out later in the year, but obviously all the way through, because it's through our network of excellence, we've had those results kind of internally as well. And they will just keep informing, what does it actually mean to implement something at scale in a mainstream service.
So Paul, what aspects of the Progress study have you taken on board regionally and locally then?
Yeah, so we had a couple of sites that were involved in the study. We unfortunately may not be taking or carrying on the work of the study. That's we need to look at that. But certainly I think it was really interesting to see how the study worked with the GP partners and really had a strong emphasis on making pharmacogenomics accessible. So there was a really strong ethos to making the results accessible, to making them delivered into the prescribing systems, to make them really sort of focusing on delivering outputs that the prescriber could then review and use in their prescribing. And so I think the most positive thing has been how well received it's been from the sites that have been involved, and how well the pathways that have been set up in terms of testing, in terms of reporting back, have worked. It really just does show that this is something that can be done in the future. And we mentioned about the 10-year plan and the importance of getting the informatics right. We know if we're going to implement pharmacogenomics, this is the kind of thing we need to be able to show. And I think it shows it really well. You need to give the results in a format, in a way that is accessible. And if we were to even think about how community pharmacy or wider pharmacy groups could be involved. There's more around medicines optimization than just GP prescribing, hospital prescribing. That information is going to be really important. It's a known issue already in terms of accessing patient records and stuff. But actually, for me, that's the thing that really helped to be engaging for the clinicians. And I think Vicky mentioned in the first podcast that certainly patients were really positive about the study, and that's really good. That gives us a lot of confidence that we don't need to overcomplicate this, because actually people are in support of getting more information about themselves and how they might better take medicines.
It's interesting. I think that there's certainly been a bit of a shift, hasn't there, with the public's sort of willingness to be involved in these sorts of things and have their data shared, provided there are checks and balances in place, and provided it's for their good and their benefit, and they can see that benefit. I think the other thing is, well, the last thing the service needs at the minute is more work with a promise of, well, this is going to improve your workload in the future. That's a difficult thing to sell to people. But if you've got something that's actually improving their workload now and making their job easier to deliver now, then that's got to be a good thing as well. So it sounds to me like the progress study, there's been lots of qualitative information that's come out of the progress study that's been used on an iterative basis and is then being used in practice actually quite quickly. It sounds like a really, really good way, pragmatic way of looking at this. Because you can imagine, can't you, some academics thinking, well, we need a randomized controlled trial. It's going to take 10 years before we can see this and all the rest of it. And that's great, but we're going to have to wait 10 years and we can't. Sometimes you just need to get on and do things. And sometimes the science can help with that sort of real world evidence, real world data, qualitative assessment of data.
Yeah, and I would make an argument that if you looking at from a scientist the literature, there is actually quite a lot of strong evidence for some genetic variation that it has an impact, and that you can modify doses and you can have a big impact for a number of drugs. So it's about identifying those best examples and also how we implement it successfully to make sure that it's consistent and we can deliver the service well, and not just in patches where there's maybe specific expertise.
Brilliant. Okay, that's been really, really fascinating, guys. Just to finish off then, right, I'm going to give you both crystal balls, okay? One crystal ball is going to be, over the next couple of years, what sort of things can people expect to see in the world of genomic-informed medicines optimization? And then maybe another crystal ball. How do you think the service is going to look in 10 years' time when the 10-year plan has been fully implemented? Vicky, so you're within the next couple of years, what are the sorts of things we might see?
So I think, as I've mentioned, we've been doing an awful lot of work in the background anyway, and I think we're starting to see that become a lot more visible. So in line with the 10-year plan, it just so happened anyway, we were re-procuring our NHS Genomic Medicine Service from April 2026. So that will be there. And obviously a lot of the kind of good timing, the things that came through the 10-year plan will obviously be a focus as part of that. And as you've mentioned, a big part of that is building up that infrastructure so we can deliver population-based genomic testing, start to think about what that looks like in terms of, having it kind of integrated across the board. So, we'll start to see that from April as we focus that in. And then, I've mentioned quite a few things that were coming through, which I think we'll start to see more of. So, information around genomics and how it informs medicine in an accessible way. We've done stuff, we've got information in the BNF, we've got stuff from the specialist pharmacy service and implementation. Now we've got the CERSI guidelines. And I think it is about having that familiarity, having it kind of, then thinking about genomics as part of your medicines optimization process a little bit more as a routine thing. We've been working on various things for workforce. So from a pharmacy perspective, we released our workforce strategic framework 2 years ago now, and we're on the third year of that. It was a 3-year kind of plan. So we're really building up to, we've got information around genomics in the indicative curricula for undergrads and we're building that out throughout, and we're seeing, competencies coming out for more expert level, in between, starting to understand more where genomics will fit into everybody's role around medicines. I think that clarity is coming. From a prescribing perspective, I think it's still out, probably not when this airs, but for consultation, the Royal Pharmaceutical Society, again, kind of commissioned via our Network of Excellence, is looking at having genomics. It's kind of a framework to support the independent prescribing competencies. So how do you bring in thinking about pharmacogenomics into your prescribing and genomics in general? So again, another tool for people to start to be using. So I'm really, in doing things like the webinars, trying to reach as many people as possible. I think hopefully over the next few years, we'll start to see more of those examples, and things like the progress study, the Network of Excellence will continue again from April. There's some really exciting work that will happen through that, and we'll start to see more and more examples of how genomics is being applied to medicines, across the healthcare system, really making it real for people and starting to build it up. So when we get to that 10-year point, we'll be working on the infrastructure. We've been working on, haven't barely touched on it, to be honest, but we've been working on, we've got informatics infrastructure, which will hopefully integrate now with a single patient record, but we've been working on the unified genomic record, for a few years now. And pharmacogenomics will be one of the first use cases over the next few years. So again, understanding how genomics information fits into everything else. So all of those things I hope to see kind of coming out, that infrastructure basically to deliver this at scale. So in 10 years time, no one will be making a big deal about this. This will just be normal, integrated, another thing that everybody thinks about when they're talking to patients about medicines. That's what I think will happen in 10 years time.
So in 10 years time, when I'm 67, I know it's hard to believe, but when I'm 67, I will have my own gene record as a patient-held record. All healthcare professionals will have access to that. So when I go and see my community pharmacist about something, he'll say, "Oh, right, yes, I can see from your notes, Jonathan, that you're more likely to respond to this medicine than that medicine." and we can factor that into your decision making. I mean, I have to say, I do like the sound of that, provided we still maintain those general principles that we've talked about what is it that matters to you, Jonathan, here. Not doing it all by rote. It's that sort of adage of guidelines, not tramlines, isn't it? We don't want to get to the point where everyone's just managed by algorithm or by AI. It can help with some of that, but it still needs to be person-centered. But Paul, from your point of view, in terms of horizon scanning over the next couple of years, is there anything that you're waiting for with anticipation? Stuff coming out of NICE or whoever?
Very interesting, because I was going to take a very different approach from Vicky. And, when I look into the crystal ball, and perhaps this is a bit more with hope than, but certainly if you sort of, what am I thinking about, the areas that we might see some progression clinically in particular is what I'd be interested in the next sort of 3 to 4 years. I think there's a real appetite and growing evidence base perhaps for pharmacogenomics in the mental health setting. I think there's a bit of work to realise that. And that would be, if I was to put it, to look into my crystal ball, I think that's what I would be seeing. And then to look at the longer term, I think there's some really interesting work that's going to go on through Genomics England and the adult health population study, and aligning with a lot of the sort of the 10-year plan ideals in terms of neighbourhood work and that wider population health. I think there'll be a big increase in 10 years time in terms of genomics being part of risk assessment. So it would be things like instead of just having your cardiovascular risk assessed, there'll be maybe genetic component to that, those kind of things. I think that's a really interesting area. And obviously, the 10-year crystal ball, bit difficult to quite see. It's a bit fuzzy, but my instinct would be that's got a real potential for impact, and I think it lines up with a lot of other streams of work. So I would not be surprised if that's something that we might see come to fruition.
Yeah, I think you're right there. That risk assessment, the risk assessment tools that we have are great, and they do help us guess a little bit better. But it's still if you look at the science behind some of the risk assessment tools, it's a bit sketchy, some of the evidence that it's based on. It's still pretty good, and it's better than just sort of an individual guessing. But I'd like to think with the advent of genomics and all the rest of it that we can do a bit better than that. So I think improvement in risk assessment would be something that would be great to see over the next few years, wouldn't it? And using genomics for that. Can I once again thank you both so much for that? That's been really, really helpful. Again, we'll put some links in the show notes to the different resources. As I mentioned in the first podcast, we're holding a set of webinars at PrescQIPP here starting in April. 3 webinars. So please register and sign up for those if you want to know more. And certainly from my point of view, I think everyone should be putting this in their portfolio and their objectives for the upcoming year or so, to learn a bit more about pharmacogenomics and indeed genomics-informed medicines optimization. So thanks so much, Vicky. Thanks so much, Paul. And we'll see you all again soon. Cheers. Entries for the annual PrescQIPP Award 2026 are now open. The deadline to submit your entries is the 15th of May. This is open to all teams working within the NHS across all sectors. It's a real opportunity for you to share all the good work that you've done over the past year and being a chance to win a prestigious PrescQIPP Award. Check the link in the show notes and get your submissions in now.